are at CDFD, and today we have Dr. Ashwin Dalal, head of the Department of the Diagnostics, and he's a senior scientist. He would be sharing his research activities and the futuristic research which is happening at the diagnostics. Welcome to neosciencehub.
Thank you.
Could you speak about your current research activities and research at CDFD?
Yes, so our institute, that is the Center for DNA Fingerprinting and Diagnostics, was set up to conduct services for patients with genetic diseases as well as to conduct research in the various genetic disorders. So our laboratory, that is the diagnostics division, it deals with the diagnosis of genetic disorders. So when patients come to us with any genetic disease, we evaluate them clinically and then do certain genetic tests to identify what is the problem with the patient, and further once we know the diagnosis, then we are able to advise them regarding any treatment available or whether we can prevent birth of another child with the same disease in that family. So that is how we try to help the patients with various genetic diseases through our work. In addition, our research also involves in identifying new genes for different genetic diseases. So as we all know that our human cells consists of the genome, and out of the total genome, which consists of about 20,000 genes, we know diseases for only 5 to 6,000. So the remaining about 15,000 genes, we still do not know what they cause, and if we do not know what they cause, then we cannot develop any treatment or any diagnostics for them. So the first step of knowing a particular genetic disease is to identify what gene causes this disease. So that is where our research is mainly involved in, where we try to collect patients where a proper genetic diagnosis is not available, and then we collect the blood samples from the family members and do whole genome sequencing or something called as exome sequencing, where we sequence all the 20,000 genes which are present in the human genome and try to identify if there is any mutation in any unknown gene in those patients. And once we are able to find that, then we can do further studies to understand what that gene does, what can happen if that gene becomes dysfunctional and all. So in this way, in the last about 7 to 8 years, we have identified about 4 new genes which were not known earlier, and that has really helped in further research in that particular area, because now more and more patients can be identified with mutations in those genes, and then further research regarding the treatment development for that disease or new diagnostics development for that disease can happen. So in this way, we try to combine both our services activities as well as the research in this particular area to help the patients with various genetic diseases.
So your areas areas related to neurogenetics, fetal medicines. So can you just explain about it, sir?
Yeah. So the whole field of medicine actually is moving towards genetics, because whether it is neurology, then we call it as neurogenetics. If it deals with the unborn child, then we call it fetal medicine. So we have cardiogenetics. So every department of medicine, whether it is cardiology, nephrology, neurology, all of them have diseases which have a genetic basis. So whenever we study the genetic basis of those particular disorders, then we add a genetics to that. So in this way, neurogenetics forms a very important field because more than 1000 genes are expressed in the brain. So brain is a very important organ, and a large number of genes are involved in the brain development. So when we study these genes, this field is called as the neurogenetics, and a lot of our work deals with patients with intellectual disability, that is, they are not able to walk or not able to speak, and all these disorders are mainly related to the brain. So that is why our focus is on neurogenetics. And the second very important field is of fetal medicine, where we try to identify abnormality in the fetus itself, so that such a fetus is not born and the parents do not have to look after the affected child and go through all the agony and all. So we try to identify the disorders in the fetal stage, and then the termination of pregnancy can be done, so that they can plan for a next pregnancy which can be normal. So in these both areas, we try to identify the cause of disease and then further work on it to create more knowledge in that particular area.
So is there any, like, can you explain the procedure, like how you can identify these diseases?
So, so whenever a patient comes to us, so mostly these patients go to any pediatrician or neurologist, and they will evaluate them, and then they see that this is not something like a acquired disease like a infection or something, and they suspect a genetic disorder. Then these patients are sent to us for genetic evaluation. So, we do a clinical evaluation, we look at the patient, the clinical features, the history, then we do certain investigations, we review the investigations like a MRI brain, a CT scan or some enzyme assays or biochemical investigations. After that, if we have a working diagnosis, then we try to identify the cause at the genetic level. So, at genetic level, as we know, our genome is made up of nucleotides, what we call as the single bases A, T, G and C. So, adenine, guanine, thymine and cytosine, these are the 4 alphabets based on which our whole genome of about 3 billion base pair is made up of. So, we try to identify any change from this A, T, G, C. If there is any change in the basis of the genome, then the disease occurs. So, to identify that, we sequence the gene, that means we identify the A, T, G, C sequence of that particular gene and we compare it with a normal individual. So, if there is any difference between the patient and the normal individual, then we consider that as a mutation or a disease causing variant, and then that variant is used for further diagnosis in other family members or doing prenatal diagnosis when they come in the next pregnancy.
What is cytogenetics?
So, we have 3 fields of genetics that we study. So, one is cytogenetics, one is molecular genetics, one is biochemical genetics. So, cytogenetics refers to the chromosomes. So, if we look into the cell consists of a nucleus and all genetic material present in the nucleus. So, when this cell divides, the nuclear material has to be equally transferred to both the daughter cells. So, for that the nuclear material is arranged in form of chromosomes. So, chromosomes are small thread like structures inside the nucleus. So, in humans we have 46 chromosomes, of which there are 22 pairs called autosomes and one pair is the X and the Y chromosome. So, a female has 2 X chromosomes, a male has a 1 X and 1 Y chromosome. So, based on the cytogenetic analysis, that means when we analyze these chromosomes, this field of study is called as cytogenetics. So, various different diseases happen because of any problems in the chromosome, the commonest being something called as Down syndrome. So, Down syndrome is the commonest cause of intellectual disability, where the child is born with an extra copy of chromosome 21. So, chromosome number 21 is in 3 copies instead of 2 copies in the normal individual. So, because of this the child develops mental retardation, intellectual disability, then cardiac defects, hypothyroidism, a large number of problems happen. So, such kind there are many other disorders like Turner syndrome, Klinefelter syndrome that causes infertility, or Edwards syndrome, Patau syndrome. So, different syndromes are there which occur because of chromosomal abnormalities, and these are identified using tests which are involved in the cytogenetics.
So, why do these happen, sir?
Yeah, so these chromosomes have to get equally distributed in the 2 daughter cells when the cell divides, but sometimes because of problems in the exact distribution of the chromosome, one chromosome comes extra or one chromosome comes less, or sometimes one part of the chromosome breaks and goes and attaches to another place. So, all these kind of different abnormalities, many of them happen accidentally. So, most of us get normal children. When people marry, almost 95% of the people will get normal children, but 3 to 4% of cases there is a problem because of different abnormalities that happen. So, we call this as a population risk or a background risk, and this happens because of certain accidents. So, accidents can happen anytime. So, in the same way, throughout the world this figure remains 3 to 5%. So, whether it is in USA or India or Africa, you will find that out of 100 pregnancies, 95 to 96 will be normal, and the remaining 3-4 have some or the other abnormalities which happen accidentally. It may not be, but sometimes it is inherited. That means the father or mother can be a carrier of this chromosomal abnormality. Once they are carrier, they are not affected, but when the chromosome goes to the child, then the problem happens. So, that we call it as a inherited chromosomal abnormality. Otherwise, the other most of the chromosomal abnormalities are accidental, and hence they are less likely that they will happen again and again.
How reliable are biochip based technologies for cytogenetics, Is it being used by CDFD?
Yeah, so we do use that. So the need for this chip-based technologies came because when we said chromosomal analysis, it is a low resolution method. That means we can identify only abnormalities which are more than 5 million base pairs. So 5 million means 50 lakh base pairs. As I said, AT, GC are the base pairs. So if anything happens to more than 50 lakh base pairs, then only we will identify in the chromosomal analysis. But if there is a problem of maybe 20,000, 30,000 base pair, a deletion, duplication, that cannot be identified by the cytogenetics. That is why a new type of technology came, which is called as chip based or microarray based analysis. So we use this test, microarray based test, to identify the smaller deletions and duplications in the chromosomes, and that is used in patients with intellectual disability to find the cause of the disease, or also in research also to find what is the problem with the patient.
Is cytogenetics being used for discovering new drugs and personalized medicines?
Not cytogenetics, but mainly the field of molecular genetics is involved in identification of mutations where new drugs can be developed, or in personalized medicine. So a lot of new drugs are coming up where they will work only on one particular mutation. Most of our earlier drugs were based on just any particular, like diabetes is there, you have these all drugs. But now what is happening is each mutation has a different drug that is coming up, and that is why we call this field as personalized medicine, where each individual with the same disease will have different mutations and different drugs will be needed for them. Similarly, when you give a drug to a patient, every patient does not respond equally. Some patients respond very well, other people do not respond. So there must be some genetic involvement, why some people respond well and others do not respond, or why some people get lot of side effects whereas others people tolerate it well. And that slowly we are finding that it is because of our genetic makeup. So some types of variants between different individuals make us susceptible to certain disorders, or make us more susceptible to side effects of certain drugs. So personalized medicine deals with the field where each drug and each patient is considered different. So two patients with diabetes will need to be given different drugs based on their genetic makeup, and this is already in practice for certain disorders, like for example, we have a drug called carbamazepine, which is used in seizures, that is epilepsy. Now this drug, if it is given to most of the patient, it works fine. But if there is one particular variant in the gene, then if you give this drug, that patient will develop a severe side effect called as Stevens-Johnson syndrome. Now instead of helping the patient, it is going to give a life-threatening problem to that patient. So at present, if that drug has to be prescribed, then in USA or some other countries, we have to do a genetic test and then only prescribe the drug. So slowly this, for many of the drugs, this is going to come, that we have to deal with each patient differently. Depending on his genetic makeup, we have to give the drug to the patient.
What is liquid biopsy? Is it reliable?
Liquid biopsy is, so biopsy per se, when we talk about biopsy, that means taking a small piece, like whether we want to diagnose a cancer or something, if there is a swelling in the body, we take a small piece out and then we study it in the under the microscope to see what type of cells are there. So that is biopsy. What is liquid biopsy is that we look at the DNA which is present in the blood. So in the, what happens when the, whether it is cancer or whether it is, we will use liquid biopsy even for fetal abnormalities. So what happens when there is any abnormality, whether it is cancer or something, when the cancer cells die, this DNA comes out and it is flowing in the blood, in the plasma of the blood. So what we do is that we take the blood sample, separate the plasma and take out this free flowing DNA from the plasma, and that DNA is used to do the genetic testing. So that is why just like a piece is taken, here we take DNA from the plasma, and hence it is called as a liquid biopsy. So it is mostly being thought that it will be useful for in cancers, it will be useful to detect even very low level cancer or early diagnosis of cancer. So even before we are able to see the cancer by MRI or something, we can detect it by doing a liquid biopsy. So it's still not yet completely reliable, we are still moving towards it, but that is something in future that is going to help us to identify cancer very early. Similarly, another very important use of liquid biopsy is something called as non-invasive prenatal diagnosis, and that is already now routinely used. So, here what we do is that when the fetus, that is the baby, is in the mother's womb, so small amount of DNA from the baby, it comes into the mother's blood. So, at present when we do any prenatal diagnosis, we have to put a needle in the mother's abdomen and take the amniotic fluid. So, that is a very invasive procedure, and many parents feel it may harm the baby and all that. So, this particular test where we look for baby's DNA in the mother's blood. So, we take mother's blood and check the fetal DNA, and through that we try to identify problems in the baby. So, that is called as non-invasive prenatal testing, and that is already come into practice, and slowly in future we will see the same thing happening for cancers also. So, it is really going to be a very important investigation in future.
So, is it harmful, sir?
So, liquid biopsy, that is the advantage of liquid biopsy, that it is not harmful, it is non-invasive. Just you have to take a blood sample instead of putting in a needle to take a biopsy, take a piece. So, taking a piece of tissue is always more problematic than taking blood. So, easily you can take blood sample and identify.
What kind of genome sequencing happen at CDFD?
Sequencing is a very important way in which we can look at our DNA. So, the DNA is very submicroscopic, you cannot visualize it with even the biggest of the microscopes. So, we have to rely on sequencer to identify the A, T, G, C sequences. So, sequencing can be done at different levels. So, the initial sequencing, what was called as Sanger sequencing, it is done to sequence small strands of DNA, like maybe 1000 base pair of something you can sequence. But our human genome consists of 3 billion base pairs. Now, if you have to sequence all 3 billion, you cannot use a methodology which can sequence 1000 at a time, that will be a lot of work. So, that is why a new technology was developed, what is called as next generation sequencing, where we can sequence whole genome together at a time using the latest instruments, and that has really revolutionized the way we can look at the various genetic diseases, because now we can sequence all 20,000 genes in humans at a time. So, for that with funding from Department of Biotechnology, we have established a center called as National Genomics Core, where we are providing services for sequencing of various organisms, right from humans to bacteria to plants to trees. Different kind of sequencings are done, and they are done for various purposes. Like in humans you will do to identify diseases, in plants they do to identify better varieties of plants to get better yield from the agriculture. For trees they will do for conservation of the trees, for bacteria, fungi they will do to develop newer treatments. So, in this way a lot of applications are there for sequencing, and our National Genomics Core provides services for anyone throughout the country to make use of this facility for sequencing their genomes.
Could you speak about S-gene RTPC test kits?
COVID-19 has brought up this whole debate about RTPCR and all. Now almost everybody knows about RTPCR. So, different genes of this virus, that is the COVID-19, different genes have been selected so that you can identify presence or absence of that gene to say that a patient is positive or negative, and that has been routinely used for almost last 2 years since COVID has come. We are using this technology to identify the virus. So, depending on where is the particular mutation in that particular variant, we can identify by doing RT-PCR, or a better method is to sequence the whole genome of the virus. So, that is what we do whenever we want to identify the variant, whether it is a Delta variant or the new Omicron variant. Then we have to sequence the whole virus, and depending on which mutations are present, we can classify it as a Delta variant or a Alpha variant in the virus. Okay. S-gene RT-PCR specifically was developed to detect a particular variant. The normal RT-PCR which was started initially, that time our aim was not to identify the variant, our aim was to just identify the presence or absence of the virus. So, just for presence or absence, 2 or 3 genes were selected from the virus and the probes were made. So, when people wanted to detect whether there is a particular variant present or not, then these new techniques are developed which are again RT-PCR based, but they will detect that specific mutation which makes it a Omicron variant or a Delta variant. Then it will be a S-gene RT-PCR. Otherwise, a normal RT-PCR will just detect presence or absence. It will not tell us the variant. But to identify new variant, you cannot do this S gene RT-PCR or something. You have to sequence the whole gene. Then only you will know a new variant is coming or not.
So, are you using any diagnosis related to Omicron?
Omicron specific diagnosis we are not doing. So, what diagnosis we do is basically a general RT-PCR based diagnosis to detect presence or absence of the virus. Then depending on whatever is the guideline from the government, depending on whatever samples are coming from outstation people coming in. So, those samples are sequenced by gene sequencing, and then we try to identify whether a particular variant is present or not.
Which area would you recommend the young researchers to choose?
So, that is very important that young researchers come into this field because many of our young people mostly go into engineering or finance, that is much more lucrative, but science also is a very exciting field. There are a lot of discoveries happening. The whole field has changed over the last 20 years. Now we have all the technologies which are available throughout the world. So, we are no way different from any other laboratory throughout the world. It is the question that more and more people have to come into this field, and as bright minds come into the field, we have seen what can happen. Like we are the country who developed 2 vaccines for COVID when the COVID pandemic came. So, there is a lot of possibility, but we need more and more people to get into it, and for young researchers there are so many different opportunities. You could either go into research field or you could go into the industry. So, a lot of industry is also developing around various genetic drug development, genetic diagnostic development. So, opportunities are there both in research as well as industry in this field, and I would encourage young students who are interested to come forward into this field and see a bright future.
Could you speak about your research?
So, our research in our lab, as I said, we have been involved from beginning to identify the mutation spectrum for different diseases in India. So, when we started almost about 13-14 years back, we did not know what kind of mutations are present in Indian population, and it is seen that whichever disease we see, the population spectrum of different mutations is very different from what we see in West than what we see in India. It's because of the genetic makeup and because of like we Indians marry Indians only, so the gene pool is different from the Caucasian, and a lot of articles and whatever data is available, it is from the Caucasian population. So, it is very important that we have an Indian database, and that helps us a lot. Like if you take an example of common disease like thalassemia. The thalassemia is a disease where blood production is defective and the child needs transfusion every month. So, if you look at the mutation spectrum in India, if we look, only about 5 mutations account for 99% of the patients. So, if you just check these 5 mutations, you are able to identify 99% of the patient. But if we did not know these 5 mutations, then we would have to sequence the whole gene, and that takes lot of effort, time for the patient. So, similar way we are trying to identify mutations in different other diseases in Indian population, and over the last 12-13 years for almost 10 diseases we have identified the mutation spectrum and published those results, and that is helping us, and that will help in future also, that any other new patient that is identified with that particular disease, we can look at those mutations which are common in Indian patients. So, that is one way, one area of research that we have been working, and we are still continuing to work. Another area is, as I said, we try to identify new genes, and in that way we have identified 4 new genes for different genetic diseases, and many more are likely to be identified. We have multiple laboratories in India just like ours who are, and we collaborate with them to identify more and more novel genes for these diseases. Another major area that we are working now is to develop a population based database. So, what happens is that when we find a particular variant or a change between the normal genome and the patient, that change could be a normal polymorphism or a disease causing mutation. Now, to identify that, we need to know whether it is frequent in the population or not. So, what is a polymorphism is that every person is different because of the changes in the base pairs among them. So, if you take your genome and my genome, almost at 1 million places it will be different, but those are called as normal polymorphisms. They do not cause any disease, they make us different from each other. So, to differentiate, when you find a variant, you have to differentiate whether it is a normal polymorphism or is it a disease causing mutation. And to differentiate that, it is very important to have a population database, like in Indian population, which are the normal polymorphisms. If we have such a database, then we can easily look at it and say, okay, this is found in Indian population, it is not disease causing. So, for that, to develop a population database, we have databases lots for Caucasian population, but for India we still did not have a database. So, we thought that we will put together the exomes that we have done at different centers in the country, and recently we put together about 1500 exomes that we had done from our lab as well as from lab in Goa Medical College, Manipal, in AIMS, New Delhi, and in SGPGI, Lucknow. We put together all these exomes and published a Indian, this is the first time we published a Indian database of genetic variants, which was made freely available to anyone to use. So, that is the first step, and soon we are going to have a much larger database through a project called as Genome India, where we are planning to sequence almost 10,000 human genomes, and that is likely to come out in the next 1 year or so, and that will be very helpful to do this kind of analysis where we want to identify whether it is a normal polymorphism or a disease causing variant. We will be able to easily identify. So, hopefully when that comes, so that is happening through 22 institutes throughout the country through funding from Department of Biotechnology, and we are hoping that when it comes, it will be really helpful for all the diagnostics laboratories.
How different is CDFD Diagnostics from other?
So, CDFD, when it was established, actually there were very few labs who were doing this kind of genetic diagnostics, because genetic diseases are rare, they are not very common in population, and hence it does not make economic sense to do genetic testing for the private laboratories, and hence very few laboratories at that time were doing any genetic testing. So, that is why the institute was established to provide services for these genetic tests for the patient, because no other labs were doing that, and slowly we have gone on adding different tests. And so, now over the last few years with the next generation sequencing and all, many private labs also have started offering these tests, but the advantage with doing with us is that we are mainly working from the government hospital perspective, and so the cost of tests in CDFD is almost 2 times less than what it would cost in private. So, in order to help the poor people where they cannot afford the expensive investigations, we provide the tests at a much cheaper cost, and ours being a research institute, we are able to do something more also, like if we find a variant, we can further study it and then try to see how it can help the patient.
So, how can they approach this diagnostic center, sir?
So, we have a MOU with a hospital here in Hyderabad called as the Nizam's Institute of Medical Sciences, which is a tertiary care hospital. So, we have established a medical genetics department there through a memorandum of understanding, and so we have clinical geneticists who are available there. So, whenever a doctor refers a patient to the clinic, the patients are seen in the clinic, then their samples are brought here to CDFD. We do the tests and then the reports are sent back. So, genetic counseling is done for the patients in our department there, and even prenatal diagnosis is offered through the same department.
Thank you, sir. We have got a lot of information from you regarding diagnostics at CDFD.
Thank you.